Okay, my name is Valerie Uytterhoeven. I'm a postdoc slash senior scientist at the VIB Institute and KU Leuven in Belgium. And today I will show you our results on presynaptic tau and how we modulate presynaptic tau pathology. So in Alzheimer's disease, you have different stages. And the preclinical phase, we see that A beta levels rise, that tau gets phosphorylated, detaches from the microtubuli, and that tau becomes cytosolic. In the mild cognitive impairment, we see that the synapses become dysfunctional, and that tau aggregates and spreads. So tau mislocalizes in the neuron in this stage and spreads to different compartments. And it would be very interesting to find molecular defects in this stage to find the cure for Alzheimer's disease, because in this stage, this synaptic dysfunction is reversible so that we can prevent neuronal death. So we are using the fruit fly as a model for Alzheimer's disease. And we used fruit flies that we developed that overexpress human tau. And we see in these fruit flies that overexpress human tau, that tau accumulates at presynapses. It was already known that tau accumulates at postsynapses, but we also show now that tau accumulates also at presynapses. Here I show you an example of flies expressing a tau P301L mutant. And here you see that tau, can you see my cursor? No.
Yes, we do. Yes, we can see your cursor.
Here, yeah. Now you can see the laser pointer, yeah. So here tau accumulates at the boutons of the neuromuscular junction, and tau colocalizes with presynaptic synaptic vesicle markers, here shown in red for CSP. So here's a zoom in on synaptic vesicle marker, CSP, and tau distributes in a donut-like shape like the synaptic vesicle marker. And this indicates that perhaps tau can interact with synaptic vesicles. And to test this, we performed different assays, a co-sedimentation assay, a co-immunoprecipitation assay, and we also used electron microscopy to image purified synaptic vesicles with purified tau. So here in this co-sedimentation assay, we see that wild-type tau and mutant tau co-sedimentates with the synaptic vesicles that we added to this assay. Also with the co-immunoprecipitation assay, we see that tau co-immunoprecipitates with the purified synaptic vesicles in the assay. And here at ultrastructure level, the purified tau localizes very closely to the synaptic vesicles here in darker shade, so that tau is very close to these synaptic vesicles. So these experiments show that tau can interact with the synaptic vesicles. But what is now the effect of the interaction of tau on synaptic vesicles? We were thinking about tau can form these tangles, so maybe the synaptic vesicles get clusters and they become immobile. And to test this, we performed a fluorescence recovery after photobleaching assay. And in this assay, we fluorescently label synaptic vesicles. So here you see a button containing thousands of fluorescently labeled vesicles. And in this assay, we bleach a small part in the buttons. And we follow then the recovery of fluorescence over time. And as you can appreciate here from the quantification, different tau mutants, the P301L, the V337M, and the R406W tau mutant, the recovery of fluorescence over time is much slower compared to butons expressing no human tau or wild type human tau, indicating showing then that synaptic vesicles in tau mutant neurons are less mobile compared to synaptic vesicles in control butons. And this also affects the neurotransmission. So the reduced synaptic vesicle mobility also reduces neurotransmission in these tau mutant flies. So here the tau mutant flies, when we stimulate the neuromuscular junction at 10 Hertz, the neuromuscular junction cannot sustain neurotransmission over time compared to the controls. They can sustain neurotransmission over time. So mutant tau has a negative effect to sustain neurotransmission at 10 Hertz stimulation. So we know that tau can interact with synaptic vesicles, but what is the interaction partner of tau on synaptic vesicles? So to find out what the interaction partner is, we performed a massa spectrometry on, with purified synaptic vesicles that we incubated with tau and then lysed and sent for mass spec. And we identified synaptogyrin 3 as a possible interaction partner. And synaptogyrin 3 is expressed on synaptic vesicles, and it's a 4-transmembrane protein, and it's widely expressed in the brain. We don't know much about synaptogyrin 3 and its interaction partners, because the knockout, for example, of synaptogyrin 3 has not much, has no clear phenotype. You have to knock out other synaptogyrin 3, synaptogyrins and synaptophysins to eventually see an effect on the synaptic function. But we know now that synaptogyrin 3 can interact with tau. And we further tested this interaction by showing in the tau mutant neurons, when we lower the levels of synaptogyrin 3, we see that tau do not distribute anymore in a donut-like shape, but distributes more widely over the pre-synapse, so indicating that the tau synaptic vesicles interaction is lost. And also with the co-sedimentation assay, we performed again this assay with purified synaptic vesicles from flies that are a knockout for synaptogyrin 3. And we see that less tau protein co-sedimentates with the synaptic vesicles. We also show that the reduction of synaptogyrin 3 in tau mutants, tau mutant flies, has a positive effect on synaptic vesicle mobility. So here the dotted line now shows the fluorescence recovery over time is rescued and comparable to control butons. And also the neurotransmission in tau mutant flies with reduced levels of synaptogyrin 3 show rescue of the defect in neurotransmission at 10 Hertz. So to summarize this first part, so pathogenic tau detaches from the microtubuli and accumulates at presynapses, where it sequesters synaptic vesicles, make them less mobile, reduces neurotransmission. We know that synaptogyrin 3 is the interaction partner of tau at synaptic vesicles, and lowering synaptogyrin 3 levels in neurons rescues the presynaptic defects. So it rescues, it lowers the clustering of synaptic vesicles so that they are become available in the synaptic vesicle cycle and to release neurotransmitters. So I showed data in flies. We went a step further to an in vivo mouse model. So we generated synaptogyrin 3 knockout mice to cross them to tau mutant mice to see an effect, the effect of lowering synaptogyrin 3, if it has a positive effect on memory and cognition, on neurotransmission, on synaptic defects, synaptic numbers, and so on. So first we created the synaptogyrin 3 knockout mouse because it was not available. So we used the CRISPR-Cas9 technique to introduce a stop codon in the second exon of the synaptogyrin 3 locus. And these mice are happily running around. They don't show any defect with body weight. Also, the synaptic markers are present. They do well in a rotarot test. Their survival is comparable to their wild-type litter mates. And here from the immunostaining of hippocampi from wild-type and synaptogyrin 3 knockout mice, you can see that the synaptogyrin 3 expression in the homozygous knockout is completely lost. So we use the tau P301S mouse model generated in the lab of Virginia Lee. And these tau mutant mice, they show synaptic dysfunction between 3 and 6 months, memory loss between 6 and 9 months, 12 months we see neuronal death, and they die prematurely between 12 and 18 months. And as you can appreciate from the immunostainings, the tau species, they spread through the hippocampus, and we see first this tau accumulation at the CA3 mossy fiber synapses in the hippocampus. And when we isolate synaptic vesicles from these mice, we also observe a dramatic increase of tau protein on these synaptic vesicles compared to wild-type mice. So we crossed these synaptogyrin knockout mice to the tau P301S mouse model, and we tested synaptic plasticity. So we induced LTP, and the induction of LTP, there is no induction of LTP in the tau P301S mice, but when we lower synaptogyrin 3 levels in the tau P301S mouse model, we can induce LTP, and it's sustained over a period of 19 minutes. So, and we performed this LTP experiment at these CA3 mossy fiber synapses where tau accumulates in the beginning of the spread of the tau through the hippocampus. We also tested the effect on memory and cognition. So we know that in the tau P301S mice that they have defects in a working memory test in a Morris water maze. So they need more time and they have to swim more to find the platform back. But when we train tau mutant mice with reduced levels of synaptogyrin 3, we see that they perform as good as the wild-type litter mates, so indicating that reducing synaptogyrin 3 levels on the synaptic vesicles leads to a reduction of tau with synaptic vesicles and more healthy synapses. We also looked into the synapses. So in the Tau P301S mice, we see at the CA3 mossy fiber synapses in the hippocampus that synaptic markers like synapsin and nectin-3 are lost. But in the double mutant, the Tau P301S with lower synaptogyrin 3 levels, we see that synapses are not lost and that we can nicely observe synapsin staining and nectin-3 staining. Also at ultrastructure level, we quantified the size, the area of the mossy fiber boutons in the hippocampus. We see that in the Tau P301S mice, this area is reduced, significantly reduced compared to wild-type littermates. But in the double mutant, so with lower synaptogyrin 3 levels in the Tau mice, we see a rescue of these mossy fiber boutons, indicating that the lowering of synaptogyrin 3 levels has a positive effect on synaptic health. So to summarize this part, so lowering synaptogyrin 3 in the P301S mouse model preserves synaptic plasticity, reverts working memory defects, and rescues synapse loss. However, we see in this Tau double mutant with reduced synaptogyrin 3 levels that Tau continues to accumulate at pre-synapses, at these CA3 mossy fiber synapses in the hippocampus. And yeah, I believe that if Tau continues to accumulate at these pre-synapse in the long term, this will also have a negative effect on synaptic function. So that's why I'm also looking into mechanisms in pathways to reduce Tau at the pre-synapse. And I'm specifically looking into the pathway of endosomal microautophagy, because we know Tau is a target for endosomal microautophagy. And endosomal microautophagy is a process at the late endosomes, and the protein HSC70 plays a very important role in this process. So HSC70 recognizes proteins, cytosolic proteins, with a KFERQ motif, so with an endosomal microautophagy motif, and Tau has two of these motifs. So the HSC70 protein then brings the proteins with the KFERQ motif, including Tau, to the late endosome, where the protein is engulfed and sent for degradation to the lysosome. And I've shown in the past that we can increase endosomal microautophagy by the expression of HSC74. And we showed in fruit flies, so we developed microautophagy sensor. And this sensor is a split venous attached to KFERQ motifs that reconstitutes when both parts are coming together, so that it can fluorescence then on the late endosomes. So I see an increase of fluorescence of this microautophagy sensor when we overexpress HSC70, and also when we overexpress chaperone defective HSC70. So we know that the chaperone activity of HSC70 is not important in this process for endosomal microautophagy. But when we express an HSC70 that is defective in endosomal microautophagy, we cannot increase the fluorescence levels of this microautophagy sensor. So we tested if the increase of endosomal microautophagy via the overexpression of HSC70 can reduce the tau levels at the pre-synapse. And indeed, when we express the wild-type HSC70 and the chaperone defective HSC70, we can reduce presynaptic tau levels, but not when we express the microautophagy defective HSC70, we cannot reduce the tau levels, indicating that the reduction of tau at the presynapse here is a result of increased endosomal microautophagy. We also see a positive effect on the synaptic vesicle mobility that was reduced in the tau mutant neurons. So we can restore the fluorescence recovery over time back to control levels when expressing HSC70 wild type, and also HSC70 chaperone and defective HSC70, but not when we express an endosomal microautophagy that HSC70. And this rescue of synaptic vesicle mobility also has a positive effect on neurotransmission. So we can restore the reduced neurotransmission in these tau mutants when we express HSC70 wild type. We can restore the neurotransmission back to control levels, and also with the chaperone defective, but not with the endosomal microautophagy defective HSC70. And so in adult fly brains that express tau, mutant tau, we can observe neurodegeneration. And neurodegeneration in fly brain is quantified via vacuoles. So neurodegeneration appears like vacuoles in the adult fly brain. And here you can see already a very big vacuole here at the arrow. And here, these upper parts of the brain, they have tiny, tiny holes. They look like a sponge. So in total, we have an increase of this vacuole area in the brain of adult flies expressing mutant tau. And this neurodegeneration can also be rescued when we express wild-type tau and chaperone, wild-type HSC70 and chaperone defective HSC70. So to summarize, the last part is increasing endosomal microautophagy in tau mutant flies, reduces presynaptic tau levels, rescues synaptic vesicle mobility defects, restores neurotransmission, and prevents neurodegeneration. So to take now the next step is to check the relevance in human neurons. So we are now checking if the expression of HSPA8, it's the human homologue of HSC70, can increase endosomal microautophagy. And indeed, again, using this microautophagy sensor, we can observe an increase in fluorescence when we express HSPA8, indicating that we indeed can increase endosomal microautophagy in human neurons. And we will test then further the increase of endosomal microautophagy via the expression of HSPA8 than in tau mutant human neurons. And to end, I want to show you that we are also busy with developing a possible therapy for targeting synaptogyrin 3. So we are developing antisense oligonucleotides against synaptogyrin 3, and we found a couple of them that can reduce synaptogyrine 3 levels in human cells in vitro and also in mouse cells in vitro. And now the next step will be to inject these antisense oligonucleotides against synaptogyrine 3 in the mouse brain and test the effect of these antisense oligonucleotides on memory and cognition and synaptic health. Okay, I want to end with thanking a couple of people. So first of all, Pablo Largo Barrientos, the PhD student in our lab that performed most of the in vivo mouse experiments with synaptogyrine 3 knockout mice and the tau mice. I want to highlight our collaborators at VIB and KU Leuven, especially Bart de Stroper and Joris de Wit, for their expertise on Alzheimer's disease and synaptic function. I also want to thank our national and international collaborators, Professor Ilse de Wachter at the University of Hasselt for her expertise on the tau mutant mouse model, Tara Spire-Jones for sharing human brain tissue, but I could not show the results of these experiments today. Also Celeste Karg from Washington University, who shared with us tau mutant iPSCs. And I also want to thank the funding bodies like the Stichting Alzheimer Onderzoek in Belgium, Alzheimer Association, and the Geneeskundige Stichting Koningin Elisabeth in Belgium, who funded these different research projects that I presented here today. Okay, thank you for your attention, and I'm ready to take your questions.